Hi, and welcome to the Anti-Waffle Podcast with me, M. Belsham-Naylor, and Marvel Gregorio D'Souza.
Yay!
And together we will try to talk about research without any gobbledygook. Each episode we'll be talking to a researcher who will discuss the work they are doing, looking at why research is important, and asking how the hell did they get into this field. We hope that it will be informative and interesting and bring a bit of the academic world into the real world. This week we have Dr Jo Santini, who is a reader in microbiology based at University College London. Jo, thank you for being here today. Could you start by telling us a bit about your research? Thank you.
That I'm a microbiologist, which you've already said, and I'm really passionate about microbiology, all sorts of microbiology. Since I got my PhD in 1999, I changed research fields, still microbiology, but I started working on a subject that is more relevant for people, and that is working on bacteria that metabolize or eat arsenic as a food source. And the reason why that's important is that more than 140 million people across the world drink water with arsenic in it. So the idea was to isolate an organism that can metabolize arsenic, convert it from one form to another that's less toxic, and then you could remove that less toxic form from the water, so for remediation. I went to the Northern Territory in Australia, which is in Central Australia, and collected some rock samples that have got arsenic in them and brought them back to the lab and isolated this gorgeous bug, bacterium called NT26, and it metabolizes arsenic. And at the time we were going to use it for cleaning up drinking water, but then we didn't do that. So instead, over I don't know how many years, 10 years, more than 10 years, I studied various aspects of how it eats the arsenic. So it eats the arsenic and gets energy from it, like you would get energy from eating a chocolate bar. It loves it, absolutely loves arsenic. All it needs to grow is arsenic air, which contains oxygen and carbon dioxide. That's it. And so I studied various aspects of how it actually eats the arsenic. And I guess that's led to more recently using the enzyme inside the bacterium that does the metabolism as a biosensor for arsenic, so as a way of detecting arsenic in drinking water. So even though we didn't use it for remediation, which was the initial idea, we are now using part of it for detecting arsenic in drinking water.
So, to make sure I've understood it, and forgive my ignorance, but how does the arsenic get into the water in the first place? How does that happen?
That varies depending on where in the world the arsenic contamination happens. So in some countries it's anthropogenic, so it's due to the mining activity, but in countries like Bangladesh, where a third of the population are drinking water with arsenic, it's natural, naturally present in the aquifer.
Oh, okay.
Right. I can tell you exactly how it gets into the water if you want to know, but that's probably enough.
That's exactly enough for me.
I was going to say, so would we have arsenic in our water? Okay.
Actually, so there are parts of Cornwall where people are drinking well water because they've got their own wells, and there's no legal requirement to check if you've got arsenic in your own well water, and it's not a government requirement. So the government's not going to pay. Water bodies aren't in charge of that. Your own private well. So there are people in Cornwall drinking water with arsenic in nature.
Okay, and so
and parts of Europe as well, but it's happening in Cornwall.
So how much would you have to drink to make yourself ill? I suppose that depends on obviously where you are and the volumes of arsenic in there. But
don't give her exact figures, just in case.
There are lethal doses, and of course arsenic was used as a poison, especially by women a long time ago. Women tend to kill people, not ex-husbands, but unfaithful husbands, by putting arsenic in their water. So arsenic was commonly used as a poison before you could detect it. It's less commonly used now, but it still is being used by some people. Not me.
Not me, by the way.
But you can kill someone. So lethal dose, you can give them milligrams per litre, is a lethal dose. But people in Bangladesh, they're drinking, I don't know, from much less than that. I was going to give you real numbers, but much less than that. But they're always drinking it. So if you stop drinking it, you would have the damage that's done, but there wouldn't be any extra damage.
Right.
But if you continue to drink the water with arsenic, then there's internal damage, and basically you end up resulting in lots of different diseases. Cancer is the most common, cardiovascular disease. It's just almost everything.
Right.
And also, if you wash in it, you end up with various lesions on the skin as well, which is often a sign of arsenic poisoning.
Oh, okay. So it's not just ingesting it; you're just getting it on yourself as well.
So was that the trigger of looking into it? Because
Yeah,
there was a lot
so
of
people
high
noticed in
number of
Bangladesh,
people.
the medics noticed that people had these symptoms of poisoning, and then they went and checked the water supply and found out this about 10 years after they dug the wells. So originally in Bangladesh they were drinking surface water, which contains organisms that cause cholera,
Right.
and various other E. coli and things like that you don't want to drink. But then they put in tube wells, which are free of these pathogens, but contain arsenic instead. But it wasn't until about 10 years after they put in the wells did they notice that people were suffering from arsenic poisoning. It takes about that long to really show symptoms.
Wow.
So your work is to identify if there's arsenic in water, is that right?
Okay, so that's the most relevant application of what me and my group have been doing is now to sense arsenic in the water. So to have a simple to use test, like a dipstick, like glucose sensors that you can buy from Boots. You put a drop of glucose on a little strip, and you put that strip into a little meter, and that will tell you the level of glucose. This will tell you the level of arsenic. So you have these disposable strips that are less than like $1 per strip, and you just throw them away after you've used them. You don't throw out the meter. You keep using that, but you could have like a meter and I don't know how many in each village, and then the strips would just be cheap and disposable, to at least tell the people that there is arsenic in their well, or there isn't, and monitor what happens over time, because there are fluctuations. So it's not just relevant for Bangladesh, but it's relevant for everywhere around the world.
So do you think that, say like one day further on down the line, that we would take these, because you found a bug that eats arsenic, and so could we just drop those bugs down in the well?
Yeah, you could. There's been a lot of reluctance by people paranoid about bacteria, because they think all bacteria are bad.
Right.
So there's reluctance by people, individuals. Just to me, it's an obvious, easy solution to use bacteria to remediate, and they use bacteria for remediation of oil spills and things like that.
Right.
So, but I think this is because it's drinking water. People are a bit paranoid because they don't understand. So it's a lack of understanding that most bacteria are lovely and good.
So you're developing a sensor which people can use to monitor the arsenic levels in their water. So that's great. And the other side is you identified this bug that eats the arsenic. So where else are you going to go? So what else are you doing with your bugs that can it just eat arsenic, or have you identified other
So we've got
metals or other bugs?
Isolated lots of different bacteria that can convert the different forms of arsenic from one form to another. So they all metabolize it. Also, one thing that I've worked on, which is finishing up now, is using bacteria for microbial mining. So this is leaching of ores, essentially. So if you've got copper or gold, a precious metal, that's often associated with iron or sulfur, and often both. And you can use bacteria to eat the iron or the sulfur and make that copper or gold more accessible to then extract. So this is for low-grade ores. So normally, the bulk of the metal is just taken normal kind of mining practice. The lower grade ores, you can use bacteria to make that metal accessible for them, the mining process, for them to then extract that gold, copper. So that's another thing we've been doing.
So it kind of eats away the
basically
dirt and stuff that's around it.
Dirt, the iron, and so it's associated with various minerals, not rocks, minerals.
Right.
And the microbes, certain microbes, these live at really, really acidic pH. so pH 1.5, if that means anything. So they metabolize the iron and or the sulfur and make that metal accessible.
Right.
For
Okay.
then chemical methods to extract the gold or the copper.
Right.
So that's what I've been doing there. But my most recent project is that where the world's, going crazy about the microbiome, the human microbiome, so all the microbes that live in you or on you. I've decided to look to see if people that are consuming arsenic contaminated water, so people in Bangladesh, have got bugs, microbes in their gut that make the arsenic either more or less toxic. So that's what we started out to do, and I just thought, well, of course it happens in the environment out there. So in the environment, there, if there's arsenic, there are microbes that convert it from one form or another to another. They can either make it more toxic or less toxic. So I thought the same would happen in the gut. So we collected, well, I didn't collect, some fecal samples from Bangladesh, human, were sent to me in the post.
Not
the normal post. There's a special courier service.
Very
good courier service.
Amazon delivering human poo for you.
Fecal samples.
Sent it to the wrong address.
No, your neighbour's got your poo.
They turned up here at UCL, and so then I went about extracting the DNA from those fecal samples, which was an interesting job. And then using a technique that my collaborator at University of California, Berkeley, Jill Banfield, developed. Basically what she does is you then sequence the DNA. So you get the DNA sequence and you get it in little small bits like a jigsaw puzzle. Right. And then she puts this puzzle together and identifies the different bacteria, the different viruses in that sample. Right. So it's like putting a jigsaw puzzle of the Rosetta Stone together. Hopefully not that complicated, but something really, the bits, and then building something, and then you get the different, all the information on the different bacteria in there. But the coolest part of this is that, okay, so there was nothing, in terms of the arsenic story, there was nothing really interesting, but this is where science is pretty cool, that you start off doing one thing and then you find something completely different that's even cooler than what you would have found in the first place. So what she found was she identified, together with her technician, identified some bacterial viruses in there. Right. Huge bacterial viruses in these human fecal samples.
Wow.
Really big, like biggest reported for any human or animal. Right. And they don't eat. So these bacterial viruses kill bacteria. Right. So a long time ago, bacterial viruses called bacteriophage were used as antimicrobial agents. So instead of using antibiotics, in the Eastern Europe, they would use these bacteriophages because they couldn't afford the antibiotics. Right. And so that's come back now, and people are looking to use viruses of bacteria because they don't kill the human, they kill the bacteria. Right. Or, they don't kill the animal either. They're looking to use those instead of antibiotics because we're running out of antibiotics, so something that specifically kills a particular bacterial pathogen, a virus that specifically kills a bacterial pathogen is a good option. Right. So there's a particular host of this bacteriophage that shall remain nameless that is prevalent. So going back a bit, so your diet will determine what organisms you have in your gut. So if you have a high, it's one of the things that determines probably, what I think it's the most important thing that determines what you've got in your gut, and obviously your health status as well. But if you're eating a high fibre diet, you're going to get certain dominant organisms, but if you're eating a high fat diet, you're going to get other dominant organisms, and so that varies between populations. So there's something about these Bangladeshis, about what they eat, that is providing the host for that bacteriophage, that bacterial virus. So it's you got a lot of the bacterial virus because you've got a lot of the host of that. Right.
Do these bacterial viruses only target certain other bacteria?
Yes. So the viruses of bacteria, viruses of animals, humans, can either be very narrow in their host range, so they'll only target one particular species, humans or a particular bacterial species if you're a bacterial virus. But there are some that go across the species barrier.
Okay.
Like, avian flu, people can get it. So there are swine flu, people can get it. You have slightly different symptoms, but there are some viruses that will infect both, like more than one species, and others that infect just one.
Okay.
And same with bacterial viruses.
Cool. So that's interesting. Can target. Specific.
So you could use specific ones that target certain species that are pathogenic, because you don't want to wipe out all your bacteria, because your bacteria are doing you wonderful things. Yeah. And you'd be dead without them.
We wouldn't be here without them.
No, you wouldn't. You wouldn't. Actually, you wouldn't. No, you wouldn't.
Be
Because you really are a bacterium. You're a living nice incubator for lots of different microorganisms, not just bacteria.
Because that could be the real world. The microorganism could be the real world, and we're just
a vehicle.
Like the planet.
We're just a vehicle for moving them from one place to another, aren't we? And making sure they can carry on. Yeah. Oh, if you think about it too much, though, it makes you go creepy. Okay, well, getting back onto it. Go on.
So back to the bug. The one that, or the big one you found, the big virus you found. Is it like a bigger version of one that's in all poo, or you're not allowed to say, or is it completely different?
I think it's completely different from anything that has been either seen or identified so far. We're trying to figure out why that is, and we're trying to look more widely at where it is in terms of other people, different people around the world, and also whether animals have it as well.
Oh, interesting. Very interesting.
So how did you get into this area of research? Did you always want to be a microbiologist?
No.
And how did you start out in your career?
So when I was little, I wanted to be a zoologist.
Cool.
So I was fascinated by animal behaviour, in particular birds. So I used to breed canaries and had all the Gould posters, posters of birds. So, and I thought primates were pretty interesting as well. But I think I wanted to be a zoologist and study animal behaviour. And then I used to collect animals as well. Not dead ones, I didn't kill them, but I used to have pet lizards and pet ant farm. And
you're like Jared Darrell.
I get a bit. If I had the space, I tell you, I would have. Yeah, so animals have always been a big passion. The thought of being a vet would have depressed me, so I wanted to be a zoologist. Not conservation biologist, but just a conserving habitat type of zoologist. And also animal behaviour is really interesting, like human behaviour. It's so cool to just sit in a cafe and just watch people.
Yeah.
The same as any other animal. It's just really interesting to watch animals behave. And so it wasn't until I started university, and then in first year I still wanted to be a zoologist. And then second year, I wanted to be a geneticist, and was fascinated by human genetics. And then I kind of became a microbiologist. And I think it wasn't, because the most inspiring lecturers I had weren't necessarily microbiologists. I think I just then found out more about microbes, and then I got hooked. And
do you think it's because they don't talk back?
Are we going to say something about your personality? Animals or microbiology? Or people?
I still love animals, and I still love birds, and got about 20 bird feeders in the backyard, and go bird watching and things like that. Why? No, it's not because they don't talk back. It's they do make me teary. This is a bit weird, but. Like, if I look, so the bug that I isolated is called NT26, the arsenic metabolizer, really, really cute. It's got, under the microscope, it swims really, really fast, and it's got these really long flagella, like Mr. Tickle, and it moves in a really interesting way. Anyway, every now and then I make it.
The bug Mr. Tickle.
No, he's called. Well, that's his unofficial name. His official name is NT26, but he's actually going to be named after my collaborator Jill Banfield when he gets properly named this year sometime. So I guess it's just that microbes. So they do make I do watch their behaviour under the microscope. But they're amazing. They're amazing little machines. They're little things that just eat different things, and they're really diverse in terms of what they metabolize, and they can live in really extreme environments, and they're just lovely.
It does seem.
I love them. I think they're amazing.
It does seem like a more.
I still love animals.
Or rather, it feels like a less discovered world, and so maybe there's more to discover, and it's more.
Well, it's hard, I guess. So you think of when I was a kid, why I wanted to be a zoologist was I probably used to watch David Attenborough, and it was just in all the the ape programs, even the butterfly programs, whatever. And so it's always on TV, and that's an obvious, if you like biology, it was an obvious thing to become a zoologist because you always see it on telly. But there are no equivalent programs on microbes, and I think there should be, and you could show the microscopic world, and some of these environments that these microbes live in, hydrothermal environments, these hot steam coming out, or obvious environments that have been contaminated by lots of poisons where there's nothing there because it looks like bushfire or something that has gone through it, with high toxins and low pH, just killing everything in its path. Yeah. But then the microbes will be there. So it's undiscovered because it's harder to study. Yeah. In that they're microscopic. Yeah. So you have to use a lot of DNA-based techniques to be able to study them. But it's, I think, much more amazing than the animal or plant world, personally. Yeah.
So go on then. Tell the people why this work is so important. Why is it so important to the public, to me and Marva?
Okay, so the arsenic research is obvious because detecting arsenic in drinking water is really important. But I think understanding the microbial world is important. Because it is an unexplored. Well, it's not unexplored, but it's not as explored as the world that we see, and it's having such an impact on so many different things that you just don't know about it, like the impact of the microbes in your gut. You don't even think about it, but without them, you really would be dead. And all the good ones in your gut are protecting you from all the pathogenic ones, or the ones that will kill you. So you need them in there, and so taking antibiotics, things like that, could destroy that happy life of the microbes in your gut. It's important for people to understand that not all microbes are bad, and actually the percentage of microbes that are bad are tiny, and some of them are just some are pathogens that definitely will not necessarily try and kill you, but harm you. Others are what are called opportunistic pathogens, and they're just bugs, microbes that are in the wrong place at the wrong time. I mean, their aim in life is not, they normally live out in the soil, but if you're immunosuppressed or something, they might harm you if you get them in your lungs, for example. But most microbes are doing really good things for you. I mean, where would we be without microbes? Apart from us humans, where would we be? We'd have no alcohol. We'd have no cheese. We would have no chocolate. No cakes.
Well, what's the point?
You're right.
Yeah, and also
very, very important, dear public, that we have this.
Also, I guess the use of microbes in microbial mining. Now they're using microbes to generate energy, so to produce hydrogen and things like that.
Wow,
so it doesn't have to be bacteria. There are microscopic algae that are microbes. So I think that we really couldn't survive without them. So we need to appreciate what they do, and I think understanding what they do. It's important to understand what they do. So it's important that we, as microbiologists, try to explain to the public the importance of microbes. And also explain to the public that you don't have to be over vigilant in cleaning because it's not necessary. It's always good to wash your hands after you go to the loo, but that's completely different to wiping down your surfaces with these antimicrobials. That's not necessary.
Okay. Is it because it kills too much, or it's not actually doing anything?
It will clean the surface. I actually did this program for Channel 4 last year, a program called Super Shoppers, to compare different cleaning products, toilet cleaning products, to see whether one worked better than the other, because there was an expensive brand versus a cheaper brand. And we found that, so it was proper experiments. So we clean well, I didn't clean the toilets, but I got samples. I got samples before after toilets were cleaned. Obviously, before toilets clean, lots of different bacteria that reside in your gut really are the most of the ones there. Then after you clean the toilet, that you can't detect anything. But then a couple of days after you've cleaned it, unless you clean it again, they're still there. But a lot of these organisms in that toilet in the first place are not pathogenic.
Right.
So unless it's something like norovirus that you picked up or somebody has picked up, most of the organisms in there are not going to kill you anyway. I wouldn't suggest you drink the water, but because that wouldn't be good. But they're not going to harm you. So and in a lot of these cleaning products are bad for the environment anyway. So you could essentially use soap water. Or not do it as often because it's not necessary, unless you have an outbreak of some major gastrointestinal problem, unless you end up with major problems with diarrhea or vomiting from something like norovirus. There's no need for you to be over the top cleaning.
Right.
Okay. And the surfaces as well. So you clean the surface, but the next day it's not going to be clean again. But it's not essential. So I think they've shown. Now that there's a huge increase in the number of people that have got allergies. There's an increased incidence of people with allergies, and people are saying is one of the reasons that people are being over cleanly in terms of not letting their kids go out and play in the dirt, being like everything's wiped down. It's so
you could be too clean,
too sterile. That's one of the reasons people think that there's an increase is because people are exposed to fewer allergens.
So they don't
just
build
in
up
early
resistance.
life, probably in utero as well, and not developing. All I'm saying is that most microbes are good, and you don't and do amazing things, and you don't need to go killing them all off. And if you want to learn more about microbes, you should go to the Eden Project's Invisible Worlds exhibition that will be opening on the 25th of May and be running for 5 years, I think.
Brilliant.
So everyone wants to go to the Eden Project, but they've got I think it's a 5-year exhibition called On the Invisible World. So it's mostly the microscopic world and what they're doing, what microbes are doing. And at the same time, if you're ever in Battersea, there will be a public art display from late summer, public art display which will represent the microscopic world of Battersea Park on 2 walls in an archway across the road from the park.
And does it have your micro?
So this is where I worked with an artist called Amalia Pica, and she's the artist, I'm a microbiologist, and so we went and had a field. We did a field trip to Battersea Park, collected samples from the ponds, various parts of the park, then brought the samples back to UCL and used different types of microscopy to visualise what was in those samples. So it'll be the visualisation of those samples on the wall.
Brilliant.
Exciting.
Thank you very much, Jo. Thank you for your time, and thank you to everybody for listening. We'll put links up to Jo's website on our website, which is www.antiwafflepodcast.wordpress.com, and on our Twitter account, which is @antiwafflepod. Please do get in touch because we'd love to hear from you. Thanks for your time. Bye.
Bye.
See, my guy's got my poo.